Good afternoon. Can you hear me? I don't know if people can hear me. You can't? Now you can? Okay, very good. So good afternoon. Welcome to the Ideas Lab. My name is Jens Eckstein. I'm an investment partner with the Hevolution investment team. I've been investing the last 20 years, mostly in the therapeutic side, but I was always interested in data. Of course, we have now the age of AI. Everybody talks about it. The question is, do we actually all know what we are talking about? And are our expectations really right of what AI can do? And I think this session, wants to deconstruct this a little bit. And I'm really happy and honored that I have two people here who have thought about AI and data a lot. They will come on stage and give a little bit of a background where they're coming from. And then I think we'll try to come up with some questions to synthesize their worlds, and then I will open it up. If you have any questions about what AI can do in Healthspan, I'm happy to take them on. So I think I will start of introducing Vijay Pandey. Vijay is the founding partner of Andreessen Horowitz BioHealth Fund and managing partner there. But he also has a long history in thinking about machine learning, thinking about data in hardcore science initially, but now it's expanding. And I've asked him actually to give a little bit of an introduction, what the history of machine learning in AI really was, where did it all start, and kind of end of today and point to some things that we know are actually really working very well and actually really have already started to change the way how we're doing science. So Vijay, why don't you come up?
Hi, so good afternoon, everybody. So as Jens asked, I'll talk a little bit about the history of AI, but first I want to put it in perspective, because actually this is something that really is a fantastic time for us to be alive and seeing. When I think about where we are today, it reminds me a lot of these famous set of pictures from the 1920s. There was a parade in New York City where they took photographs every year. And in 1920, the parade was primarily people on horses. And then year by year, it was more cars, fewer horses. And 10 years later, it was basically all automobiles. And that doesn't sound all that impressive to us today, but you have to think about what happened over that 10-year time scale. Basically, the modern world was created, a world that started with horses and no electricity, to the modern world of cars and electricity and all the things that we take for granted. And that's the analogous transition we're going into today, from a world without AI, where we have just sort of the ways we're familiar with doing things, towards building the new modern world, where AI will be everywhere the same way that electricity is everywhere now. And so, maybe to give some sense of where we are in that arc and where we've come from, what AI is basically the ability to predict. That it's a mathematical means that takes data in, learns patterns, and then predicts what comes from that. And this has gotten more and more sophisticated over time, but actually is many years old. 10 years ago, 20 years ago, this would be typically called machine learning. And in machine learning, what happens is that a human being comes up with a problem and then feeds in data that it figures out and then tries to make predictions from that. It could be something simple like, can you measure someone's cholesterol and predict the risk of heart attacks? Or in other medical areas, maybe putting in some information about a drug and predicting whether it's going to bind to a protein or something like that. What's gotten us excited today is the scale in which this learning can occur. And so what we see today with large language models is that we can now put in not just a little bit of data, but put in the whole sum of human knowledge. So we can put in everything from the internet, everything from medical textbooks, and again, we're predicting, but what we're predicting here is something that maybe sounds odd from a prediction point of view, but we're predicting the answers to questions people may ask, predicting the words that's going to come in the answers. And so in a sense, it was really, the foundations of this are very old, But at scale, something really magical happens. That these current generative AI can answer questions and make predictions of things that now starts to feel very human-like. And what this means for us today is, just as we have this transition from horses to cars 100 years ago, we're going to see roles for this that will replace the types of things that we would normally maybe expect a human being to do. This will start in maybe simple tasks with helping you write a document or read a document and so on, but we're already seeing this applied to nursing and areas in preclinical care. We're seeing it applied to small molecule drug design, and I think in time, it'll be very intriguing to see what areas get impacted. I think the one thing that has always been a common sort of belief in technology is that, oh, with this technology, all of us will be out of a job. And actually, when people talked about factories, they actually talked about this idea that we would have 20-hour work weeks because the machines would be able to do so much. And as I think each of us can attest to, unfortunately, I don't think too many of us get to have 20-hour work weeks. And so I think the irony, though, is that this actually won't take away jobs. It'll take away some types of jobs, but it's going to create new jobs. And especially, I think the exciting part is allow us to do the things that are perhaps the most exciting and interesting. And the more menial, the more sort of boring everyday things that AI can do today would be things that I'm very excited not to do. In terms of then sort of how to think about it and where we are, we're kind of in the beginning of this arc. Like in this 1920 to 1930, we're in the very earlies of the 20s. That, you know, we're used to horses, but you see a few cars. And for those of you that are maybe suspicious of AI or maybe not as exuberant as I am, actually, this is what happened with cars as well. In the early days of horses versus cars were actually not that great. Like horses didn't require maintenance. You know, they can make their own horses from horses. You know, you don't need this infrastructure of gasoline and all these things. In the beginning, cars actually looked not really all that exciting compared to what you could do with horses. But I think similarly, AI has its limits today. But those limits, and this is maybe where it's different from cars, are actually decreasing relatively quickly. And I'll leave you with one of the biggest exciting areas, which is that you've probably heard of Moore's Law, the idea that compute is actually decreasing in cost by a factor of 2 year over year, or increasing in power by a factor of 2 every year. That's still occurring. And so the hardest thing for us to really, I think, internalize as human beings is what that geometric growth looks like. A factor of 2 doesn't sound all that impressive, but a factor of 2 times 2 times 2, 10 times over a decade, that's sort of a factor of 1,000. And so these computer models that cost, let's say, $100 million to make today, that's a huge amount of money. In 10 years, that would be $100,000, which is not a huge amount of money. If an AI doctor in your pocket might cost $20 today to do, or maybe let's say even $1,000 today, that'll be $100, or $1, sorry, in 10 years. And I think that's the part that really gets us especially excited, is that so many things in our lives, when we think about healthcare, but also education and other areas that we need professionals, those things are increasing in cost exponentially. And the ability to jump from that increasing exponential to the decreasing exponential, that is perhaps the big fundamental change. And the economics of a lot of things that we're used to will go upside down. For example, like all of us would love to have a medical professional with us, let's say all the time. I could imagine someone looking at the food I eat or the work I do, or maybe I'm feeling a little sick and they can make recommendations. But very few of us could afford that. And especially in a world where we want to move to health span and prevention, that would be something that could really greatly facilitate that. But the economics don't work out with the current costs of professionals. But if AI can take over those tasks, these things become actually available such that things that were economically unfeasible become feasible and even advantageous, especially since preventive medicine could save us a lot of cost and grief in our futures. And so what I think we're going to see is both a change in terms of how work is done, in terms of AI versus people, but even just the work that we can choose to do. And so I think in this arc 10 years from now, perhaps if we rejoin here 10 years from now, I think we would be actually very much at the end point of that, where right now we're talking about AI, it's very hot. My big hope for everybody 10 years from now is that AI gets to be very boring. That we're all used to it, it's all in our pockets, it's not a big deal. It's as boring as the next iPhone release. Like, nobody cares about that because it's all become very powerful technology that we're used to. And I think that's the promise of where we are, but it's the beginning. And so in terms of Hevolution, I think this world where we can have health span prevention actually I think is nearing, and AI will play a huge role in that.
I mean, the one thing you said is Moore's law, that confusion speed gets higher and higher and higher. I think what's really important to also understand is the question you're asking, right? Because the question has to somehow fit what you can do on the computational side, right? I mean, if you think, for example, about protein folding, right? When I went to grad school, we still get crystallography and, we had to do all those things. And then we ran some simulations and it took forever. For one simple question, you had to put little boxes around, only look at this part. Now 20 years later, we have computing power that can actually do a lot of those things. Yeah. So I think the question, the expectation you have for any given problem needs to be somehow attached to what computation can do at any time. And I think that's where very often there's a disconnect today when people say, well, AI can do that. You really have to ask exactly, what is your question? Yes. To really understand whether they can do it or not.
Yeah. And towards that point, I think the biggest issue is, does AI have the data to be able to do that? And so much of everyday things are on the internet, but actually most of healthcare data is not on the internet. And the key part of healthcare data is, for us as individuals, that my personal data, hopefully, is not on the internet. And I think that's going to be the key learning that AI needs to get into. But also, I think the other area in healthcare and medicine is that perhaps the most interesting things aren't even measured yet. And so measuring things, new things, that type of data set that AI could learn from is particularly exciting.
And just for complexity, I mean, if you would compare the complexity of folding of a 400 amino acid protein with the human biology, I mean, are they on the same level? Are there, what's your, give us a
so and actually, I've spent a fair amount of my career in protein folding as well. And so, I mean, the reality is that we're comprised of so many proteins that that's just the beginning in a sense. But I think the ability to predict protein structure, what's exciting about that is that it's a problem for which there was a lot of data into a very challenging biological problem, and now is basically a solved issue. And I think it's a paradigm for what we could do beyond that.
Yeah, very good. Okay, great. So I bring you back, and I want to go over to Jamie Haywood now. Jamie Haywood is an engineer, so he's trying to engineer biology and looks at biology very, very different. He has a long history, looking at data. He started out with data that was mostly wrong and tried to figure out which one was the right data and the wrong data. But I let you tell your own little career path. That's very interesting. And then really come from the data side. We ended up with the data that's out there. You have spent so much time on data. Give us a little bit of feel what kind of data is out there, what the issues are with data, what other data we really need. So start from the beginning and then broaden it out from there.
So I think it's worth starting with a little bit of bias about AI. In order for us to learn all of the world's knowledge, we had to digitize all of the world's knowledge. And in order for Google to do that, Google had to go and put readers in every library in the world and turn the pages and OCR the pages of all the books in the world. And they did that back in 2002. And that was a form of AI, machine learning of characters, a little bit like AlphaFold. It gave us an understanding of a language that we could then begin to use for something, but we did not have the computing power or the ability to distill it into the representation of human knowledge that we have in the language models today. And I think when I talk about bias, and I love these models, we use them natively in all of our work, but one of the reasons they're so impressive is because they encapsulate all we know. But that's all they encapsulate. They encapsulate all we know. And so when you go to the data that is missing, and I'll go back to sort of my early career in ALS research, I had a very selfish motivation. I wanted to help my brother. I wanted to find a drug that would help my brother. And so I tested every single idea ever suggested by anyone in the world in a mouse model of ALS to help my brother. I spent $25 million over 5 years, and I was unable to replicate a single study by anyone in the world. I'll repeat that. Unable to replicate a single study, including studies that led to human trials that killed people faster. And this became part of the replication crisis. And so what was the point of that? Some of the data we think we have is not accurate. Some of the data we think we have is biased and is informed by human beings with motivations and things like that. So if I go back to the question that I think Jens just asked, what is the problem we're solving? I think in Healthspan AI, we should be ambitious. And AlphaFold is a miraculous piece of software. The ability to impute genetics with AI is miraculous pieces of software. We use these daily in our work to enhance things. The ability to design antibodies with these new tools is enhancing all of our work. But the sum of the parts tells us very little about a human being. Each protein has 10 to 20 epitopes in it that could be phosphorylated, glycosylated, nitrosylated, or other things that we have not yet measured on them. And so that's 2 to the 10 different variations, and that's one protein that AlphaFold can fold once without some of the nitrosylation to one other protein, not the, how many in a cell? Trillion in a cell, give or take? So the layers of what we're doing right now with biological AI, we're just reading the words. We're just beginning to read the words. But if you want to go to understanding this, I think you have to realize you have to generate data. And this is what I came to in my first project in this area 7 years ago, where I actually just had a simple question. What data plane, genomics, RNA, proteomics, metabolomics, lipidomics, antibody profiles, cytokine analysis, cell surface markers, was predictive of all human variation? Most predictive. What predicted BMI, what predicted whether you had diabetes, what predicted whether you had ALS, or multiple sclerosis, or fibromyalgia. So I ran an experiment where I recruited in the United States about 2,700 people. I drew blood on them longitudinally. Some of the blood was in the same day, some of the blood was 1 day, 1 week, 3 months, a year. And I ran all of these data planes, and I compared them, not to see if one biomarker was worthwhile or not, but what the sum of all the biomarkers told me. And from that came out the sort of the learning that proteomics was the most stable and predictive of human variation in the time frames I care about. And I want to talk about the time frames I care about for a moment. So HbA1c, lipids, blood pressure, and viral load. So lipids have added about 2.3 years to human life in the United States of America. Not by one drug, not by one action, but partly by banning trans fats, but by learning how to get rid of the entire consequence of excess lipids, if you choose to, on human bodies. So when we have a biomarker that is stable and measurable, we fix it. It takes us about 40 years, but we fix it. HbA1c, we can now do the same thing with the GLP-1s and the other drugs for doing that. And essentially, if you choose to, you can prevent the entire consequence for 90% of type 2 diabetes. And hypertension can also be managed in the same way with either lifestyle, behavior, or drugs, to the consequence of being hypertensive is gone. So when we have an actionable biomarker that stably reflects someone's future, we can fix the problem. So to me, the problem is, how do we generate the biomarkers to fix the problem? And the group that did the most work on this was the UK Biobank, that around the same time period when I was doing my study, Rory Collins began this large-scale program to collect plasma over time. He realized that state biology was the center of the universe. For all of its flaws, it is without question the best longitudinal study of human beings ever done. And last year, a group of pharma companies published and released proteomics on 50,000 people from the UK Biobank. And in our hands, and not just our hands, that proteomic release alone explained half of the unexplained variation of human beings. So in other words, if you add up the entire history of medical knowledge, we took 50% off the thing in November of 2023. And we're doing 600,000 people now. So I think we're entering an era where we have potentially, for the first time, the data. I think I want to go back to the language models and the sort of the generalization, because this is not something that's going to get fixed by just dropping in human knowledge on top of this. This is going to require thoughtful, careful analysis of how you do this in an integrated way. It's going to take the integration of the language models to explain the response variable characterizations. It's a significant engineering project. But the project outcome can be a set of biomarkers, the Star Trek of health, that say this is health. And and my red number 2 should be higher, or my green number 4 should be lower. And if we get ourselves into the right thing, just like we did with lipids, HbA1c, and viral load, which took a universally lethal disease from 8-year death to a normal lifespan, biomarkers cure disease. Drugs follow the biomarkers, so if we make them, we create them, that's how we will create longevity.
BJ, maybe come up on the stage as well. So I wanna stay on the data part a little bit, because I think what a lot of people don't understand is how important the data is. I mean, there's the old saying, input, determines how good your output is. And I think that the big question is always, is there enough data today to do anything about human biology? And if yes, what is it? And if you want to create more data, what is the data that we really need to create? Can we prioritize in one shape or form what kind of data we need to collect? And any of your opinions is valuable here.
I mean, the data creation has to follow the problem. And I think that we have not done enough thoughtful analysis about what makes an effective clinical marker. And if you really look at how they're used and how they vary, their sort of stability in the context of short-term variation, understanding the properties of what making something a really good clinical marker then determines what data you want to collect. And I think the data you want to collect has to be targeted to purpose. And so, for example, the UK Biobank looked at 10, 15-year changes in health states on 40 to 70-year-olds in the UK. Very useful, not the entire population, and not younger people and not much older people. But they don't look at short-term variation in that data. And so the data set that I have, which was designed to eliminate that short-term variability so that the biomarkers would be useful, is what's necessary to make that other data more useful. So I think the problem and the mathematics that you're doing define the data that you want to collect, and that's an iterative process that will continue to go more and more.
Yeah, so in life sciences, all of the AI stuff is coming at the same time as a major renaissance in biology. And this renaissance is robots and automation, but also things like CRISPR and the ability to do experiments you just couldn't do before. CRISPR and stem cells and so on. And so the data generation is kind of amazing. And the two go hand in hand, because actually doing all that data generation without the ability to understand it would actually not be that useful either. And so the bio revolution's coming at the same time that AI needs it, and the same time that bio needs it. In care delivery, actually, there's something analogous, which is that, but maybe a better analogy there is almost what you see with Tesla and self-driving cars. That I don't know if any of you have seen these Tesla cars, but actually they can drive themselves to some degree. And how is that possible? Well, actually, while you drive it yourself, it's monitoring all the things you're doing, and it's learning from that. And so AI that's gathering data explicitly to learn can be very powerful. And in care delivery, you can imagine AI sort of piggybacking and watching what a doctor's doing and learning from that. And along the way, as the doctor's working, it's not just helping patients, but it's training the AI. And so the companies that are the ones providing the most successful and helpful care delivery services could become the companies that are the biggest AI care delivery services in the future.
And do we know which data is good data and which data is big data? I mean, it comes to the question always of, do you actually have to clean up data before you use it, right? Because you can imagine you just have, like a lawnmower that just, eats all the data that's out there, and, you feed it in there, and then you hope something, comes out of it, and you get a model. I mean, how much time should we spend on actually understanding the primary sourcing data?
Yeah, well, here in drug design, actually one of the bigger issues is that, at least in the past, these campaigns weren't done to train AI, they were done to come up with a drug. And so that data is probably not the best for AI and requires a fair amount of cleaning. Whereas now the data is done explicitly to build a model. And often actually AI, through what's called active learning, can actually tell you what data it needs to be able to learn better. And so that whole idea of data generation for AI is something very different than what we had before, and that's intrinsically cleaner data.
And I think that the one other thing, the flip side of the data collection and the model building, that is really the validation, right? And depending on where you're coming from in the world, the term validation can mean something completely different, right? I come out of the drug discovery and development world. For us, validation is actually a signal in human, right? Yeah. That you can measure, right? Validation is not an enzyme assay. And I think that's where the big question is: how far along are we, for these new models in human biology to actually be the same as a clinical signal that I actually measure in a human being? What do you think? Where are we on that journey?
Yeah, so one thing to think about is, like, where's the bar? So you actually mentioned ALS in mice. For certain diseases, mice models are accurate. For others, actually, they're not very good models at all. And naturally, we can't experiment on human beings. And so AI as a model, I wouldn't expect it to be perfect, but actually it just has to be, in my mind, better than the animal models for us to be able to do something materially better. And actually, it's for many diseases, it's already there, especially in diseases where, let's say, cell phenotypes can recapitulate the disease phenotype. And many companies are taking advantage of that to identify new targets and even to try to run and design clinical trials in a more effective way.
Carefully, and then the AI can do amazing things, but if we feed it data that is generated for biased or other billing or other reasons that don't have a lot of information, it's very not accurate. I think that gets to the, like, to me, we talked earlier about the Turing test. Like, I mean, the goal I'm trying to shoot for is to make meaningful predictions in an individual that are robust in an individual, to the point where if you change something in an individual, you take a drug, you can see the effect in the individual and trust the result against the biology that you're measuring in one person. Now that's an insanely ambitious goal that requires an immense amount of correction and data collection and integration. But I think that goal forces the creation of the data that will allow the integration of the layers, because you know why you failed, and that's what you learn each day. And I think that's what the point of trying to make an AI that's actually a real digital twin that you would trust a treatment recommendation from is something where you really can get that right in one person.
Well, I would add to that right now the process of medicine is you come to a doctor with some symptoms or some disease, and you're measuring things to treat the disease. I think with AI now, there's the opportunity to imagine measuring things to keep you healthy.
Yep.
And that's a very sort of a huge change in paradigm that wouldn't make any sense without the ability to interpret that data, and especially that data longitudinally. And that's a huge opportunity.
Yeah, and I think there's an immense amount of low-hanging fruit. I mean, again, we discovered lipids in the 60s and 70s in the Framingham Heart Study, same with hypertension. And so we haven't made a lot of advances of high-quality causally reactive biomarkers in human biology. And if we had more, a connective tissue biology set, or a biology set that looked at IgE propensity for overactivation, like all of these things that make us ill, just like lipids, every one of those, if we had them, we would be able to identify health and become healthy. And hyperlipidemia is not a disease, it's a way to prevent disease. Mm-hmm. Hypertension is the same thing. So I think we can have 10, 20 of those, and we can make them mathematically more ideal so they're independent and actionable.
Yeah. Well, one last thing on the topic is that, so I was meeting with a team that's developing one of the major AIs that you played around with, and they're a super impressive team, and they're saying that they're finding it increasingly hard to find problems that AI can't tackle. And so I was asking it, well, what are you doing in the medical side? And they're saying, well, we're having AI take medical exams, and it's as good as some of the best doctors, and soon it'll be better than all the best doctors. And I said, well, but like, where I would set the bar higher than that is that right now what AI is doing is it's learning from the textbooks and recapitulating that. What I would love AI to do is to take in all the medical information and tell me where the textbooks are wrong. Mm-hmm. Number one, and to see if you can make your own textbooks, that would be better. But then the second thing that's even stronger is, can you make a textbook for me? Because each one of us are different. When it can do that, now we've reached like an amazing level, and I don't think that's that crazy far off. Maybe it's something like 10 years.
And I think that's a really important point, because I think pretty much until today, or still today, most of medicine is based on population averages, right?
Yes.
I mean, we compared against everybody here, you know. I have to be a piece of you in order for the doctor to say something about me. I think AI really gives us the opportunity to say, forget all about this.
Yes.
You're going to be your own baseline.
Yes.
You know, I will compare whatever happens to you
Yes.
in a better state, right?
Yeah, and you can only do that though if you have data on your baseline. Right.
But building on that, like every clinical trial we've ever read, broad statement, overclaim, has 3 variables that they evaluated: BMI, age, and sex. And they balance the groups by those 3 factors, and they tell you whether the results were statistically different in those 3 factors. So there's essentially 3 borderline binary variables we use to context all human difference. I'll repeat that, 3 binary variables to context all human difference. Right now, our current factor model has 26 orthogonal vectors that explain 75% of all human variation in biology. And so those give you an independent set of numbers that allow you to evaluate or balance a group or normalize something in a clinical trial to see if the biology you're looking at is different in some group or another group. And that's what you can do with this. And that compression allows us to get far more information from the trials, from genomics, from these other layers that have been suffered from a lack of ability to show statistical significance because the variables we use to stratify people are not very robust.
So coming back to your arc of where we are on that journey, if you go to your doc today, you know, and you go to your typical yearly, what are the questions you should ask him that they should do beyond the normal, 20-something panel, everybody's doing? Are there questions that you should actually ask your physician?
Yeah, I think the shift is going to have to be, can you measure things before you get sick rather than after you get sick? And there's companies doing this, and I've invested in companies doing this. And that's such a huge shift from, at least in the US, especially how medicine's paid for. Because you would not order a test if someone doesn't have symptoms. But from my mindset, like, I think about this, I compare it to, let's say, the fire department. It wouldn't be that, like, I can only call the fire department when my house is on fire. Or when I'd like to sort of, when I have even the smallest suspicions, I'd like to be able to do something about it. And that shift towards measuring things while you're healthy, and then giving you topics to talk to your doctor. That's what's happening right now.
Yeah. So I think, there's no reason to figure out how you can biobank some of the things now, right? Because that could be a comparator, in 5 years down the road. Are there services like this where, you can get your blood frozen away, and then 5 years later you go back and you can compare yourself to the 5 years younger?
Thank you for the layup, Jens. Yeah, I mean, Alden Scientific was started sort of 25 years ago when my brother was sitting in the doctor's office, and the question was asked, how is Stephen different from other ALS patients? How am I different from Stephen? How are ALS patients different from non-ALS patients? And over the years and the different work I've done in disease discovery or drug development, I've used these different data planes. And to me, I think it's now we can do this in individuals. And I think the challenge is, if you don't keep where you are right now, you lose it forever. So if your plasma is not frozen right now, that data's gone forever. I test my blood roughly every week, sometimes multiple times a day, sometimes a little bit less frequently. But I also bank 30 aliquots. So we're going back and we're running, PFAS to look at that, or we're looking at other
I'm gonna
environmental
open it up,
chemicals that we can look at that, so we can identify what correlates would be driving or not driving health. And the idea is that there's a group of people that, for the first time, rather than waiting for research to discover something and hand it to them, we're a bespoke discovery company. We discover things with you in real time. You have all the raw data, all the raw mathematics, and that's what we do as a business. And Jens might have a few vials in our freezer.
So I would love to open it up. I mean, you heard kind of a pretty broad stroke at what AI can do in health. Any questions you want to ask over there? Yep.
You said we only know the sex, name, and BMI. Can you elaborate more on the other section, what we are able to do today, and how is that different?
Yeah, I mean, there's a lot of mathematical techniques, but one of the things that I've been interested in is what is common among humans that's measurable and different, so that when you're looking at a trial result or a drug outcome, you can look at common factors that interact with that. And there's some mathematical techniques. Factor analysis is one. There's other ways of measuring latent spaces. And so what we're doing is we're just basically doing a autoencoder for a human being, compressing them down, and then using that reduced dimensionality to ask, in some context, what's different. And that allows us to get a lot more insight into how those differences play out. And a particular advantage is in a small study. We're doing a study of chronic Lyme and long COVID. It's only 300 people, and you would not have the power in that smaller study, 5 groups of 60, to find anything unless you do some very aggressive normalization through the lens of this sort of harmonization process. And so by normalizing the group, we can essentially identify what signal is showing up uniquely against 50,000 people, not against 300.
Here's a question.
Hi, this is Anay from New. So it's a semi-question, but a semi-comment as well, related to this N of 1 sciences versus, let's say, the data aspect of AI learnings, right? So New in a way is doing what we're talking about, doing all the multi-omics, AI-driven interpretation as a SaaS, B2B-driven platform to commoditize these types of learnings, right? And so, like you said, Vijay, is now we're seeing this convergence of both the testing being sped up and the AI now getting the data to be able to learn from that. And what we're kind of really excited about our platform is, yes, we've been able to learn mostly so far based on the big biobanks data, the UK biobanks, lifelines, millions of clinical trial data, and so on and so forth, right? But it's all population data on one endpoint in a generalized population study, right? But that's as much as we know today, right? As we're moving forward and as practitioners can use this type of insight, multi-omic driven insights to guide their practice, and they're now saying, okay, I'm doing this intervention for my patient with chronic inflammation, but the other guy has a different philosophy and will take a different interventions. Now all of these people are feeding back the AI to say, okay, similar profiles, similar genetics and biology, but different interventions. Now we're learning from data how those are taking place. So within a kind of an end clinician-patient relationship that's possible, but how do you think this N of 1 trials, for us it's clear like how they're training our AI algorithms, but how do you see that from the regulatory perspective that will be able to shift from the paradigm of today's FDA RCT trials, everything that has to work the same for everyone, versus we really need to go into precision, personalization, and so on? If you have a comment.
Yeah, so there's a lot there, and I think a couple different things. One is a lot of medicine has to make the assumption that we're all very similar, and we all don't know that's an oversimplification. On the other hand, I don't know if we need to have 8 billion models of people. There could be maybe 100 phenotypes or 10 phenotypes, and those phenotypes will come up very naturally once you have enough data. And so then, as you mentioned, there could be, let's say there's 100 phenotypes, we might have still a lot of data for each of those 100 phenotypes that would be predictive. And maybe it's also predictive for how to move me from the healthy version of the unhealthy VJ phenotype to the healthy VJ phenotype and so on. And so that could be predictive and something you could do proactively. So that's something I think that's coming, and I've seen that technology starting to be put into practice. But you also asked about the regulatory aspect about N of 1 trials, and that's hard in terms of, I think the burden of proof for RCTs is still high. What could happen, and who knows, but what could happen is that, especially since even phase 3 clinical trials doesn't have as much information as phase 4 as what you get with real world evidence. There's been an interest to have a test for toxicity, and as long as it's as long as it's safe, then you could let real world evidence and precision medicine take it from there. I think we need to build up the first part to get to the second part, because I think it's hard to motivate the second part and this idea of reducing the trial sort of expectations before we can predict that. But that could be what could happen.
There's another question here.
Hey, Rakov. My question is kind of a follow-up around sort of data quality specifically. So, specifically around sort of building a lot of these models, we are seeing sort of the rise of sort of these single-cell GPT-based models and so on and so forth. A lot of these are trained on sort of publicly available data from Geo and so on and so forth. And you pointed out very correctly, the quality of this data is questionable by both of you in a lot of ways. Now, are these models that have been built, are they trustworthy in some ways or not? That's my first question. And my second question is, if these models are trustworthy, and you did proposition to sort of generate a lot of new data, that's very expensive. So is there a sweet spot in terms of sort of using publicly available data and sort of still leveraging new data?
I'll take the
But I'm going to give a specific example. So the Broad Institute has done a lot of work perturbing cells. And they've done that with RNAi, and they've done that with compounds, and they've done it with lots of different kinds of human immortalized cell lines. And when you analyze that data, if you don't do it carefully and you try to ask the question, like what drugs are similar or what drugs are similar to what genes in the way they perturb the cells, what you find is that they didn't manage the dosing very well, and that most or half the cells are overdosed. And so what you're seeing is, 5 or 6 classic ways a cell goes into extreme distress, but it doesn't tell you anything about what the drug does. And so you have to then correct for that and get rid of that. And and they've done better at CellSeq, the next generation of this stuff. But that kind of, like, what's the model being trusted for is the question, right? So, I mean, would I trust a drug recommended by that first version engine? No. Would I be interested in one that after we'd found the sweet spot of the S-curve and we started to get a confidence that we were seeing the effects of the drugs and the genes? A little more. And so that's kind of, it depends on the problem you're looking at.
So I would add that, like, if you think about the current LLMs, they just learn from what's out there. So in the medical world, it's from textbooks. And so they're almost like the best med student ever, but they don't have any practice of seeing patients. And that's the part that data is typically not available. But for companies that have that data, they could take that best medical school student ever and try to make it the best doctor ever. And the bar is not, do they never make a mistake? The bar is, how do their mistakes compare to human mistakes? Because unfortunately, we're fallible too. And I think this is going to be the general thing with AI is not, let's say for self-driving cars, do they never kill anybody? But if they, let's say fatalities are 1/10 of what it is with people, then it would be immoral not to have them because all these lives would be saved. I think when AI reaches something similar in the medical world, I think there'll be a similar desire to put it into practice.
That co-piloting idea is really important because there's a lot of evidence that, I mean, AIs are less biased, they're less likely to make sort of single source decision errors, but they don't have the experience of the individual. And so the combination of those two things, having a conversation, we do it now when we're analyzing someone's individual biology, where we'll have the physician, the member, one of our informatics teams, and then we'll have an LLM listening and bringing in research that's related to the conversation in real time. And so that kind of complement, I think, is going to produce far better outcomes. I think the other thing is we don't know what's right. I mean, like the new models are new. And you know, and I think the reason we talk about regulation is that, you know, that when you release things in the real world, you want some confidence that they're robust and validated. But when you're discovering things, you don't know which is right. You don't, like, whether the traditional recommendations are right or the new recommendations are right. And that's a conversation. That's a learning experience. I think one that it makes sense to dive into rather though than avoid.
Okay, we have time for one more question.
Thank you so much for this interesting talks. Dr. Basmat Manu from Ophthalmology Department at King Saud University. I'm practicing as an ophthalmic genetics consultant. So in daily life, I'm facing so many patients with genetic conditions. We do requesting for them genetic, whether whole exome or gene panels or so. But what is most important for me in relation with the patient, in the time when it comes with the missing heritability or, let's say, negative results, is there any way in the AI to enrich the data where we can catch up these information so we can tackle, have a smooth way to understand what is missing? Because sometimes it's very difficult to find an interesting institute with the same more or less patients, so you can start to do a real research. And you know, AI is open, so you cannot trust every single point in it. So, for your opinion.
You want to take a let him?
I think that in the next 4 or 5 years, there's going to be a significant increase in polygenic risk score interpretation. I'm not sure the medallion genetics will get that much better, but you'll find biomarkers linked to those. We build biomarkers to the genetics now. I don't know how you, I think the other question you're asking about, how do you integrate the data and connect it? I think that's a systems problem beyond my scope.
Well, yeah, I would add that, like, the AI that you see today, I wouldn't think of it as the full picture of AI, much like if you remember when Microsoft Windows came out or Netscape Navigator came out. Windows is not all of computer and Netscape is not all of internet. You need to have these other components, need the applications. And so I think you should view these LLMs as the operating system, and the applications are to be built. And that's what I think we're all collectively in the process of doing.
All right, we're out of time. Give us a hand. Give a hand to Jamie and Jake. Thank you very much. Thank you.
Everybody, it's 2:45 and I'm going to start the next session. Please don't leave. Hopefully a few more people are going to join us. So I want to talk to you today about a novel class of molecules with what I believe is incredible promise in diseases of aging that we call mitochondrial microproteins. And my name is Chassi Cohen. I'm the Dean of the School of Gerontology at the University of Southern California. It's a school that trains 600 students a year in various aspects of aging research, senior living care, gerotechnology, and other aspects of aging. And as many people have alluded to today in various talks, we believe that the ultimate prescription for maximizing lifespan, health span, and brain span is diet and exercise. And that's always been the key to healthy aging. How that happens, we're still learning. And what I believe, and one of the central tenets of the hallmarks of aging is the mitochondria, which responds in many ways to diet and exercise. And today I'd like to convince you that the way this happens is by the mitochondria releasing a new class of peptide hormones, also known as microproteins, that have important relevant activities to promote healthy aging. So our lab is in the interface of 4 specific areas: diseases of aging, of course, and aging itself; mitochondrial biology, that I'll tell you a little bit about; big data and precision health; and microprotein science, particularly microproteins that are encoded from the mitochondrial DNA, which is a little-known small chromosome inside mitochondria. And mitochondria is key to energy production. People know it's the powerhouse of the cell, but mitochondria have many important functions besides that, including regulation of cell death, immunity, overseeing metabolism, and they're a key component of the cell for healthy longevity. And importantly, mitochondria is the only other organelle outside of the nucleus that has its own DNA, its own chromosome, which is maternally inherited. There's no recombination and no admixture in mitochondrial DNA. We only get it from our mothers. And it has important elements that contribute to protection and risk for a variety of diseases of aging, as well as longevity itself. And here you can see that, as of course you know, our bodies is made up of cells. Each cell has a nucleus, other organelles, and of course the mitochondria. The mitochondria, as I said, has their own chromosome that encodes for 13 large protein-coding genes that are part of the respiratory chain of the mitochondria. But 20 years ago, we identified a small peptide that we called humanin that's encoded from the 16S ribosomal RNA of the mitochondrial DNA that has important protective actions on cells. And later we identified another microprotein that we called MOTC that has since proven to be an important element in healthy longevity. And these 2 microproteins, along with numerous other peptides that are encoded in the mitochondrial DNA, are secreted from outside, from the cells into the circulation and have important protective activities on muscle, fat, brain, heart, and other tissues. Now, here you see a map of the mitochondrial DNA, which is only 16,000 base pairs in size, but contains over 700 putative small peptides. And when I say peptides, think about the size of GLP-1, which is the active moiety in Ozempic. And 700 potential ones, we're studying hundreds of them. There are about 11 or 12 that have already been published. Chief among them, and the first to be described, is humanin, then MOTC, which has some very interesting activities that I'll tell you about. Others like Schmooze, Schlep2, we've published. And today I want to particularly highlight a new one that's not published yet that we call Mazel, which is highly relevant to people living in Saudi Arabia and the rest of the Gulf. So we discovered many of them. The way we discover the ones that have important function is using AI approaches on multi-omic analysis of large databases. And we test these to prove that they have important biological functions and look for opportunities to address diseases of aging like diabetes, obesity, and AD. And the discovery of microproteins that involved humanin in 2001 and some of the other ones that we discovered has now expanded dramatically. And we say there are 700 possible ones in the mitochondria. There are tens of thousands, if not hundreds of thousands, encoded in nuclear chromosomes, representing a new level of complexity in biology. And traditionally, people thought there are 20,000 genes in the human genome. Now we know there are hundreds of thousands if all these microproteins are counted. And these small open reading frames that encode these peptides really add complete new levels of complexity. And these are now been part of our understanding of biology for 20 years. And we believe that just like the first wave of cloning of genes 35 years ago, this represents an important opportunity in drug discovery. And every wave of drug discovery over the last century followed a basic set of understanding of biology that preceded it 10 years later. In the 1960s and 70s, we learned about enzymes, allowed us to develop all these small molecule inhibitors. When we started cloning DNA, we followed with developing with companies like Amgen and Genentech multiple recombinant proteins. The RNA era that started in the 1980s, 1990s gave us the companies that now control the RNA manufacturing. Same thing with gene sequencing. And the discovery of microprotein already involves multiple companies that are capitalizing on these for drug discovery and development. And I believe this will be a new wave in drug development. And I think particularly important is going to be the mitochondria as a source of these microproteins. There are potentially many hundreds of them. They're involved in the mitochondria, because of its involvement in aging itself and diseases of aging, seems to be an important source of relevant microproteins that have metabolic, immune, and CNS regulating activities. One of the ones that discovered is called MOTC, which we've licensed to a company called Cobar that took it all the way through a phase 1b clinical trial. It has very interesting activities. However, the formulation that was used in those studies isn't adequate to move forward, and we need to reformulate itself. But interestingly, we discovered that there's multiple ethno-specific conditions in, say, Hispanics or Arabs that allow for the specific development of those microproteins that are relevant to those conditions in those people. We discovered these using a series of omic approaches, defining the ORFs themselves, looking at epigenetic regulation, gene expression of these small open reading frames, identifying the peptides through mass spec, and using population genetics to see if there are SNPs or variations in the body of this open reading frame encoding microprotein that are associated with increased or decreased disease risk in particular populations. We also developed assays to measure the levels of these, and we're using these as templates for drug discovery. And again, The discovery pipeline always, for us, starts with genetics, using a variant of the GWAS approach, or whole genome sequencing, to identify SNPs associated with disease. We follow that with developed a unique approach to expression of mitochondrial RNA. If you will, this is an RNA-seq version of the mitochondrial RNA. We also develop a unique software to identify, using mass spec, these unannotated microproteins in MS. Then we use a variety of cell lines and mouse models to refine and identify the functional aspects of these microproteins, and then using analogging and preclinical drug discovery and development, we're trying to move these into the clinic. So here's one of the examples of this mitochondrial GWAS that we call MIWAS, where we conduct a genetic analysis of a particular trait, in this case, early onset cataract, and in this case, in a population of Hispanics. And we found a SNP that is only found in Mexican Americans, in no other population, and people who carry this SNP have 5 times the risk of developing cataract. This affects a mitochondrial protein that we haven't worked on since, that changes its structure as a result of this mutation, and therefore likely represents the cause of this early cataract. A much more interesting and well-published microprotein that was named humanin was simultaneously discovered in my lab and in Iku Nishimoto's lab in Keio University in Japan. And you can see here, we cloned humanin using a yeast-2 hybrid system before we developed our genetic approach as a partner of IGF-BP3, which was involved in diabetes and Alzheimer's disease. You can see that you can take normal neurons, add amyloid beta to them, and most of them die. And if you also add humanin, you protect these from death. We also showed that administering humanin to mice improves their cognitive function. We outlined signaling pathways of humanin. And then we discovered a mutation in the humanin gene, which is associated with reduced levels of humanin. And also, the same folks who carry this mutation with low humanin level are much more likely to have rapid cognitive decline with aging, particularly in this case for African Americans. There's also some effect on white people, but a lesser degree of an effect. Interestingly, when we make transgenic worms that overexpress humanin, they live longer, and mice that receive humanin have improved health span on many levels, including obesity, diabetes, cognitive dysfunction that's delayed. Also interesting that long-lived mice have higher humanin levels, and short-lived mice have lower humanin levels. I told you about the transgenic worm that lives longer. Naked mole rats have higher and sustained levels of humanin compared to mice, but the most compelling evidence for the role of humanin in human longevity is that centenarians, for which, of course, there's no living control group, but if we look at children of centenarians, they have much higher levels of humanin in their plasma than age-matched controls. These are all about 7 years old. So humanin is clearly a lifespan, health span regulated molecule with potential for drug development. Last year, we published that there is a new variant of humanin that we call P3S humanin, which is associated with exceptional longevity. Its frequency in the population is very low, less than 1%, but among centenarians, the frequency of humanin is of this particular variant of humanin, the SNP, is 15%, and it co-segregates with ApoE4. ApoE4, of course, puts you at risk for Alzheimer's, but also is associated with early death. So you would expect ApoE4 frequency to fall rapidly with age because most of these people die, but interestingly, it stabilizes around age 80, with about 15% of the population over 80 have an ApoE4 allele. Almost all of them also have this P3S allele, and then we showed that P3S humanin potently binds ApoE4, stabilizes it, and apparently prevents the damaging effect of ApoE4 on cell death, particularly in the brain. Then we take mice that are engineered to overexpress human ApoE4, infuse them with either humanin or the P3S humanin, and dramatically decrease the amyloid beta accumulation in the brain of these mice. So clearly this is a relevant microprotein with potential in human aging. The next microprotein that we discovered, called MOTC, is not from the 16S, it's from the 12S ribosomal RNA region of this mitochondrial chromosome. And interestingly, when you give MOTC as daily injections to mice that are fed a high-fat diet, it has a dramatic effect on preventing obesity, better than GLP-1, and even more importantly, GLP-1 analogs like Ozempic, as you know, are very effective for weight loss, but 1/3 of the weight you lose with GLP-1 is muscle. MOTC induces weight loss, but is muscle sparing, and in certain cases even increases muscle mass. It also improves insulin sensitivity in muscle of aged mice, and it inhibits the production of myostatin, which might be one of the mechanisms through which it activates muscle growth. But its most dramatic effect is absolute prevention of fatty liver development in high-fat diet-fed mice. An analog of MOTC completed a phase 1b clinical trial in people with NASH in a company that I co-founded called Cobar. And then about 4 years ago, we discovered that Japanese and Korean people carry a mutation in the MOTC gene that changes the structure of the microprotein, and the same mutation could theoretically affect the 12S ribosomal RNA, but as you can see here, this variant has no effect on the structure of the 12S ribosomal RNA, a dramatic effect on the structure of MOTC, rendering it bioinactive. And then we've been able to show that those Japanese folks in 3 different cohorts who carry this mutation have a 50% increase in the risk of diabetes. Interestingly, this only happens in men and not in women, and there are many examples in the mitochondrial genetics that there is a sex difference in these mutations. Even more fascinating is that in one of these trials, the JMIC trial, we divided the population into very active, moderately active, and sedentary, and showed that only in the sedentary group, the presence of the SNP increases the risk of diabetes. That means that if you carry the SNP, but you're physically active, you can overcome the loss of this microprotein. Also interesting, of course, as I said, that this is a male-specific phenomena. We can detect very high levels of the inactive MOTC hormones in the blood of the men who have this mutation. And in addition to being bioinactive, we can show that young men who are otherwise perfectly healthy, who carry this mutation, already have an increase in abdominal fat, which later in life leads to the development of diabetes. Now, what we think about MOTC is that on the one hand, it's exercise mimetic in the sense that you give mice or humans an injection of this, and their body believes they've just exercised. But MOTC is also secreted in response to exercise, so it's exercise-induced, and it's exercise mimetic, and it's also exercise enhancing. Here you see how long mice can stay on a wheel when they're exercising, and you see old mice and young mice. And then when they're injected with daily MOTC for 3 weeks, you can see that both young and old mice, and it's more dramatic in the old mice, are able to stay much longer on the wheel, improving their ability to exercise. And here you can see induction of MOTS-c in an acute exercise bout in obese, recently diagnosed new diabetics. Even more exciting is the fact that when we give MOTS-c, and because this is a mouse experiment that takes many months, only 3 times a week, and ideally we'd want to do this as a daily injection, but the IRB wouldn't let us do it, we actually can make these mice live about 10 to 15% longer. So this is one of those molecules that increases life expectancy in mice, improves insulin sensitivity and obesity in humans, and we think has incredible potential to be developed. The development is currently on hold, but we're hoping to be able to take it to the next level with a new formulation. Let me share about, real quick, another microprotein we discovered a couple of years ago that we call SHMOOZ. We looked for microproteins that might be involved in Alzheimer's disease. So we conducted our MIWAS, our mitochondrial GWAS analysis on people with and without Alzheimer's disease, and found a SNP that increases the risk of Alzheimer's pretty dramatically by about 50%. The SNP is found in 20% of European and 10% of Asians. And that makes it the second most important SNP in Alzheimer's disease after ApoE4. We also were able to show that when we take mice with the ApoE4, or this one is 5X-FAD mice, and we treat them with daily SHMOOZ for 2 months, you also see a metabolic effect and some weight loss, but you see a pretty substantial effect on reduction in plaque size and number of plaques that are large. So this is a potential intervention for Alzheimer's disease. And interestingly, not only is it just like ApoE4 associated with AD risk, it's also associated, if you have the mutation, with a shorter life expectancy. Here is the chance of making it to 95. If you have the SHMOOZ SNP, it's reduced by about 25%. If you have the ApoE4, which is a more potent bad SNP, it's reduced by about 35%. If you have both, it's 60% less. So this is not a SNP you want to have, and if you do have it, we have to figure out how to overcome it. We developed a novel bioinformatic technique that we call MINIT, Mitochondrial Interaction Test, and been able to identify that there is genetic crosstalk between SHMOOZ and a novel nuclear SNP that is associated with changes in life expectancy. So the SHMOOZ SNP reduces your life expectancy, but the interactive nuclear SNP can dramatically affect, and we're talking about several years life expectancy difference if you have the good version or the bad version of this nuclear SNP in the presence of the SHMOOZ SNP. We've also been able to develop an assay to measure SHMOOZ in plasma. Interestingly, SHMOOZ improves in response to healthy diet. It goes up in people who adhere to Mediterranean diet. It goes up with olive oil, and people who have lower levels of SHMOOZ are more likely to develop Alzheimer's disease independently of the SNP status. And one more quick microprotein that we call SHLEP2. We were looking for SNPs that are associated with the risk of Parkinson's disease, and lo and behold, we found a SNP in the mitochondrial DNA that doesn't cause Parkinson, it protects from Parkinson's. And what this does, it changes the structure of SHLEP2, and it's really a gain-of-function mutation that makes SHLEP2 more potent in protecting brain cells from pesticides that cause Parkinson's disease, both in vitro and in vivo. But I want to spend the rest of my time talking about what I think would be an exciting discovery for this nation, this crowd, and that's a peptide that we call MAZL. Mazel in Arabic as well as in Hebrew means fortune. And the rapid industrialization in the Gulf and in Saudi Arabia has led to an epidemic of obesity, diabetes, and the complications of obesity. And not only this is a major problem, there is obviously some genetics involved in this. And as all of you know, almost every drug that's ever been developed has been developed for Europeans in either Europe or United States. They typically tend to work relatively well in the population they were developed in, but less well in other populations. And for many diabetes drugs, there is a failure rate of about 30%. That's, for example, the number for metformin. But that failure rate is higher in non-Europeans, suggesting that we need focused, targeted drug discovery for various ethnicities for things like obesity. So this is the frequency of diabetes in the Arabian Peninsula since the 1980s to today. It went up from being quite rare to being frighteningly high in recent times. So that brings us to this new microprotein we call Mazel, which is not published yet. The only thing that you might find in the internet is the patent that we wrote on it. It's a novel therapeutic target that, again, is a microprotein produced by the mitochondrion. And we discovered it not in an Arab-related environment, but in the HRS database, the Health and Retirement Study, where we do a lot of our work, as there was a variant that's associated with more inflammation. And we started playing with this microprotein and noticed that it has effects on weight in mice with a high-fat diet. But then, scanning the literature as I do for mitochondrial variants with relevance to the phenotypes that I'm interested in, I first noted this paper that came from a group in Kuwait that showed that there is a SNP, which is haplogroup defining, that is associated with protection from obesity. And then just last year, another group looking at subjects from both Kuwait and Qatar. And by the way, if anybody here has genetic data on mitochondria available in Saudis, I would love to work and see if we can demonstrate that this has a role in additional populations in the Gulf. But they again showed that there's a SNP that's associated with protection from obesity. So when we went to the UK Biobank and found this SNP, and lo and behold, as we also noted in American databases, we did not see any association between the SNP and the risk of obesity in whites, blacks, Hispanics, Asians, and Indians. But when we went to the UK Biobank and specifically looked at Middle Easterners, which is not a very well-defined ethnicity in large population bases, we found that in Middle Easterners, it was protective against obesity. What that teaches us, and you noted that in this technique that we call MINUTE, is that in addition to the biological effects of these mitochondrial variants, which I showed you can be quite important, it's the nuclear infrastructure, architecture, and the various nuclear SNPs that exist in a population that talk to the mitochondrial SNPs that determine whether that SNP will be protective, will have bad effects, or will be unaffected, depending on what nuclear SNPs exist. So I believe that in Arabs and people of Middle Eastern descent, this protective SNP allows protection because there are other genes in the nucleus that offer it this protection ability. So here you see the protective effects in two of the published studies. We mapped this SNP that was protective to a D-loop open reading frame that encoded a microprotein that we call Mazel, and showed that this mutation changes the structure of Mazel And it's not an infrequent SNP. It's between 10 and 20% of Arabs. Those are the people who don't get obese. Other folks who don't carry the SNP in the presence of high-calorie diet will gain a lot of weight. It's protective, and this SNP may be critical in understanding the biology of obesity in the Middle East. So we developed an assay for mazel. It's an age-dependent hormone level. There's difference between men and women. When we take the wild-type mazel and inject it into mice, we see reduction in fat and some gain in muscle at the same time. So unlike GLP-1, unlike Ozempic, where you see reduction in fat, which is wonderful, but you also see reduction in muscle, this one is not just muscle sparing, it builds muscle and may have an important therapeutic effect. So this is with wild-type muscle that all of us make. Here is what happens when we take the new variant of mazel with some other modifications. It has a very, very potent effect on inhibiting weight gain, decreasing fat, and improving muscle mass in mice. And we believe that this is an exciting new therapeutic target. It is not yet in preclinical development. We hope to be able to obtain the support to do that in the coming year. And I'll finish here, but just by saying I told you about several exciting microproteins. There's several more that we're working on. There's one called MENCH, which is a mutation in Mexican Americans and other Central Americans. Millions of people carry this, 30% of them. And we believe it's the cause of Hispanic diabetes in the US and could be corrected with MENCH. We have a couple of, most of the peptides I showed you so far do good things. But we started identifying some peptides that do bad things. PATS is a peptide that's the opposite of mazel. It makes you gain weight and lose muscle. We developed a neutralizing antibody against it that we think might be useful in sarcopenia and frailty. SCREAM is a novel somatic mutation in cancers that accelerates cancer growth. Again, a target for inhibition with neutralizing antibodies or other forms. And in fact, we're working on some specific longevity-related peptides that might be actual longevity drugs when we're all done with them. So our approach is based on precision medicine discovery and drug development using multiple bioinformatic big data AI approaches. And I think these are important, potentially translatable discoveries. We managed to get MATSI into humans. We hope to get more of these microproteins into clinical trials. And particularly mazel is a compelling preclinical drug development target for the 400 million Arabs who are now benefiting or suffering from Western diet-induced lifestyle-associated diseases like obesity and diabetes. And we already have an assay that we'd love to provide for people who are interested in this molecule. And we love to collaborate. We have many, many reagents in the form of peptides, assays, antibodies, mouse models. And I want to thank my external collaborators, many noted people. A few of them are in the meeting today. My wonderful teammates, and of course, the funding support from the NIH, and particularly from Hevolution, who've been incredibly transformative for the field of aging. And thank you. Be happy to entertain some questions. Yes, sir.
Thank you for that great talk. Have you tested any of some of the mitochondrial targeted supplements like some of the NAD precursors, maybe urethrin A, mitochondrial, to see whether they have an impact on improving humanin and MOTS-c.
I would love to have the opportunity to test this. We haven't done that yet. Anybody who's running a little trial? We have done, we have a big nutrition program, and we measure these in all of our nutritional interventions. Mediterranean diet, low-fat diet, low-carb diet, low amino acid diets, and many of them go up in response to that. We have one, which we call MIS-MDP, metformin-stimulated mitochondrial-derived peptide, which is dramatically induced by metformin. We're testing that as well. So there's just so much we can do, but if anybody has a set of well-characterized plasma samples, we'd love to measure.
Hi, thanks for an amazing presentation. Could you expand a little bit upon the comments you made about the bioavailability of MOTS-c, and you referred to the fact that you have to reformulate it. I also think it's in wide use at some clinics now.
I don't know how widely used MOTS-c is. Obviously, it's not approved, and I don't sanction any use of it. I know that there are several clinics 2 blocks from my house in Santa Monica near Los Angeles where you can get MOTS-c. And I know several people who were taking it for a while. MOTS-c has a cleavage site in amino acid 8, which can be degraded very easily by a number of proteases. The version that was developed at CoBar was analoged to eliminate that cleavage, but the wild type, if you inject it in mice intraperitoneally, it doesn't get cleaved, it circulates, and it has amazing effects. But if you inject it subcutaneously, most of it gets degraded. So those people who are injecting it off-label in these longevity clinics are probably not getting much benefit. And also, even at its best, you need to inject MOTS-c daily. In some of our mouse studies, we give it twice a day. And obviously, if this is ever going to be a drug, it needs to be modified so it can be given weekly. The expectation for biologics that are used for diabetes and obesity now is a weekly injection. And even if you have extra benefits like muscle sparing, you got to compete with the weeklies. So as much as I'm admiring the phenomena, and I think most of it, when we published a paper that shows 15% increase in life expectancy in mice, everybody started talking about it. I hope some of these people would like to invest in a biotech company that will create a version that is going to be effective, safe, and long-acting. Yes?
Thank you very much. Earlier today, you heard all of the excitement about bringing drugs like this forward into clinical trials and making them into drugs. What is your biggest hurdle? Is it funding? Is it regulatory? Is it safety? What's your biggest hurdle to actually getting these in humans for clinical data so we can make drugs?
Without a question, it's funding. So we raised $80 million for CoBar, which allowed us to develop the particular analog that was more stable, a little more longer acting, but required, and this is not my area, very sophisticated formulation, which led to local injection side effects, which was a problem that required us to dilute it more to prevent that. And when we completed the phase 1b study, there was clear indication that it was working, but it was also clear that we couldn't go into a phase 2 with this preparation, which meant we had to go all the way back and re-engineer the molecule and the formulation, which was priced at well over $20 million, which we weren't able to raise. There was the pandemic. I don't know if you remember how, and you know, a company that burns a lot of money had to close down. So funding is important, but also I think that people like me come with a particular set of expertise. I'm a drug discovery guy. I also have the clinical connection to diabetes and obesity care, but there is a whole field of protein chemistry that's become really sophisticated over the last few years. You know, when GLP-1 was first identified and the first versions were terrible. And then Novo Nordisk had a fairly good daily version, and then Eli Lilly and Novo Nordisk moved to the weekly versions. There's an army of protein chemists out there that we needed on our team 3 years ago, but we didn't have them. Today, I think we could make this happen collaboratively with the kind of expertise that we're missing. So collaborative drug development, funding, and I have to say the FDA can be pretty tough about what I thought at the time were pretty minimal local side effects. They put us on clinical hold, don't ask.
Regulated under CEDAR?
Yes.
Okay.
Well, thank you all for staying, and I hope you enjoyed the talk.
Okay, so thank you everyone for joining this session. So today I will be sharing mostly about our work at Regenerative Bio. So we are AI-driven longevity company working on the organ-specific agent. So mostly I will talking about the technology platform we developed called More Clock, and also its applications in the precision anti-aging solutions for organ-specific kind of agent mechanisms. So my name is Gavin Zhou, and I'm the co-founder and CTO of this company called Regenerative Bio. So with some of the backgrounds, I think most of the audience are the experts in longevity, or at least some of the fans in the science and the longevity economics. So people have been interested in the longevity, also called anti-aging, for quite a long history, even long before the modern medicine or even the inventions of antibiotics. But within the thanks to the development of the modern medicines right now, we begin to tackling the kind of diseases such as cardiovascular disease, cancers, and metabolic diseases one by one, and begin to realize it's not that efficient to tackle these diseases one by one. That's the most of the themes discussed this morning and later this afternoon about considering aging as an entire status of your health instead of tackling this one of the each of the diseases one by one. So here you can see it's kind of an incidence for different diseases, which by means is the occurrence rates among the agents. It's very kind of difficult to appreciate this diagram as it's showing the log ratio, but it's going very fast as being aging in terms of the incidence for each different kind of diseases, especially during the age after 60s. So right now it's brings more and more attention to considering aging as more kind of risk factors or the combined kind of background for all these diseases happens. And right now I think there has been a lot of questions raised. How can we tackle aging as one of the problems? And here we show us through the understandings of aging biomarkers and the researches on the mechanisms of aging, we gradually realize that using the biomarkers of aging not only can help us to tracking or understanding the aging, but also can help us to know some of the diseases or the change of your health status in advance of time. And this is the applications on so-called predictions or the prognosis when someone developed a disease or several kinds of diseases. And during this kind of intervention or so-called treatment, it's also very important to monitoring the entire process along the way, how people are reacting to different drugs, to certain therapeutics, or even the other kind of so-called nutrition or diet, etc. And lastly, it's most of the focus and a lot of the audience and the speakers have been touched on is the novel solutions. It's not only about, say, a traditional kind of a drug, or it's more kind of a solution. How can we provide to tackle the problem of aging? So I will just dive a little bit background on the development of measuring aging, as this field is mostly graduating or growing after the technology developments on the next generation sequencing or so-called omics kind of approach. And there has been several measurements called clocks or aging clocks built to measure all this kind of different process or status of aging, and mostly are relying on this called DNA methylation. So it's one of the kind of epigenetic factors on the different genes. You can imagine it's more like the switch on different genes to turn the genes on and off and to regulate them and help them to interact with each other. And there has been mostly starting initials on the chronicle kind of epigenetics, which is connecting your age with your actual age, or called chronicle age. And later on, people just gradually realize not only these biomarkers can help you to, without looking at your actual age, to predict your chronicle age, but this can also indicate some of your risks on certain diseases, such as the diseases we just mentioned. And later on, people just gradually using more kind of advanced and longitudinal kind of study data to follow each individuals on different time points, so that we can check the changes on different and also measure the pace of aging. And this is more we've been focusing on, and this is more people will be more caring about during the kind of monitoring and the intervention kind of process. And with all these kind of developments, being able to using all these different clocks to measure the aging paces and also to predict some of the like diseases factors. And these are using similar kind of technical approaches called next generation sequencing or DNA methylation or omics data, but they are using slightly different approaches to looking at different markers. Some of them are more focusing on aging perspective, and some of them are more focused on specific diseases such as cancer. You know, a lot of efforts are on the early diagnosis of cancer. So at Regenerative Bio, we are building this kind of platforms to connect all the dots together and hoping to generate this kind of a closed loop from the measurements to interventions, and later to combine this together in a more kind of clinical or practical way. It's more in a scenario like the longevity clinics nowadays. And the platform we are building, one is called the organ aging clock, and the other is the anti-aging substance screenings. And all these are backed and powered by our AI platform. So I will start with the measurements first. Here is just a whole diagram for our measurements on the aging, and we mostly rely on the methylation as our major verticals, but also we are combining different kind of physiological data and other omics data together to better tweak this kind of measurements of aging. So in terms of capturing the signals from the DNA methylation, we need to have the like blood sample collected from the individuals or the participants of different trials or the different kind of interventions. And this will go through a more kind of from the so-called wet lab process, which is to extract the molecular signals from the blood, and later to the dry lab to dissect the signals from each different organs and later mapping them to the aging status. So I won't dive into too much details because there's a lot of like technical aspects, but the most idea behind this is to extract different kind of signals from the blood, and later the signals will tell you where it's come from different organs or different kind of systems like your immune system, and later this can be mapped back to the biological aging kind of models. And this is a more kind of a showcase on our algorithms later on to mapping different of the aging processes. So one is called the more kind of using the ideas learning from the artificial intelligence application in weather forecast or different kind of mapping. It's more about because the blood is like a mixture of different signals from different organs, but eventually we need to tell like which molecules or which kind of signals are coming from, say, your lung or your gut or different kind of systems. And this is how we dissect the signals. And later on, this will better to building this model on our kind of traditional aging biomarkers with different kind of a training process to further identify like the age of, say, your lung, like or your gut or your colon, etc. And all this data has been validated and compared with the public available data, thanks to the researchers from the all over the world, especially led by Dr. Vadim Gladishev's group in Harvard, building this BioLearn kind of sketch to combine different data sets together to help us to better harmonize and compare different kind of organs and different kind of aging clocks together. And we luckily to compare our existing kind of more clock data with the other public available and published algorithms. And this has shown extraordinary kind of in terms of the accuracy and also the effectiveness on the kind of aging predictions. And later on, we are just with all the model change, we are fine-tuning this with different kind of population backgrounds. As we all know, like from the more kind of a genetic or gene perspective, different populations have their own kind of genetic backgrounds, and also even when speaks of aging, all the factors such as diet or life habit will matters. So we combine all these like factors together with a more focus on the different populations to fine-tuning this kind of aging clocks using, say, the Asian-specific kind of population, and even into, say, different kind of subpopulations such as Chinese, Japanese, and also we are starting some of the collaborations here in the Middle East to further tweaking like this aging clocks into a more designed and specialized for different specific groups. And right now we are collaborating with two of the top hospitals in China, namely the one in the southern area and one in the northern area, to combine this together to building a 3,000 people cohort to further tuning this model with a baseline study. And the other part is about interventions. So before I dive into that, we will just start a little bit background on the current or so-called state-of-art kind of deep neural network prediction on the senolytics. As we all know, there has been some of the well-known kind of aging intervention perspective. Namely, we all know metformin has been a very hot topic, and also some of the audience discussed about the NAD plus pathway or the other well-known kind of aging pathways. But there's more than that. And we are hoping to leveraging the power of AI to help us identify and finding more kind of a powerful and safe and effective kind of senolytics or so-called anti-aging solutions for different aspects. And this is some of the existing work done by a group in MIT, and they have shown like within the help of deep neural network, we can already identify and using the existing data sets to further predict and to finding new kind of senolytics or so-called anti-aging solutions. And right now, there has been a technical problem is most of the models are just using very partial of the data, namely the like the atoms of a different structure or the just simple the chemical bonds of different structures. But combining all this together and even providing this context to the model has the meaning and eventually will help with the so-called the large language model, the LLM, behind all the well-known like ChatGPT or DeepSeek, can help us to providing a better context and eventually reaching a more kind of precise and effective way for predicting this kind of anti-aging solutions. So right now at Regenerative Bio, we are building this kind of approach, as I mentioned, to combine the existing kind of a diffusion model together with a multi-view model and with the help from large language model together to help us better scoring this kind of molecules and also finding the new molecules. And this will go through some of the algorithms. I don't have time to dive in details, but again, this will helping us not only to finding existing or repurpose some of the molecules, even the natural compounds and the drugs we already have today, and to finding their applications in the anti-aging dissectors. And also, the more powerful and the more bright future of this is about to helping us to learning the new features and even finding the molecules which has not yet been synthesized, but with the more kind of potential on the anti-aging kind of applications. And this will eventually go through a more kind of large volume and high performance, high throughput validation platforms building in our in-house to further validate all this with the chemical and powers thanks to the automations and all the high throughput kind of screening methods developed today. And right now we have been reaching some of the existing data to show our model on the pure dry lab hand or pure computational, and has the power to finding a more kind of synergetics within the same kind of training and validation data sets, just like trying to compare apples to apples. And with all this, we can show not only the accuracy, but also the eventual AUC is the overall kind of taking into effects of the power to finding the right synergetics, but also to finding the effective numbers of all these synergetics, is to showing we can reach better performance with all the kind of existing models currently available. And with that, we are further validating this with the kind of a well approach, thanks to the developments to the organ, the organoids, which is more like an organ on a chip to further help you to understand the 3D structures and also the microenvironments of different organs. So here we use some of the kind of a heart and also lung and ovary as different organoids to test the synergetics. And here we just show some of the preliminary data from the heart organoids, and it already show some of the power force. And one of the molecules is the kind of positive controls we use is the P16, and the others have been showing is the novel molecules we already found. And there's more to come within the data generated and more kind of the data provided by our automation and robotics platform. And this is just a showcase on the kind of our AI screening platform. So we will focusing on some of the specific organs, and it's targeting the health needs, relying on the understanding of its pathways or the potentials. And we will start with some of the biologicals and the understandings of different targets and pathways, and combine this with the kind of comprehensive approaches with more combining the biologicals from the physiological or the understandings from the clinical perspective. And later, the current approach is mostly on the safe ingredients, which is under the kind of FDA approved, whether it's already approved drug or it's more kind of a natural compound. And later, the animal experiments will be performed, and some of them will even be collected at the cellular or organelle level. And eventually, the clinical trials will be applied to further validate these solutions together. And eventually, we will have this kind of composition. So some of them can be novel molecules, as we mentioned, and this will go through the traditional kinds of IND and drug discovery process. But again, if it's kind of approved or already existing kind of molecules, it can go through some of the fast tracks in terms of the repurpose, or even go through the supplementary or the nutrition kind of a perspective to go into as an actual solution. So right now, we are performing one of the clinical trials on the ovarian anti-aging kind of a process, and this is designed to measure not only the aging clocks we already have, but also the kind of physiological the indicators from a more traditional and clinical perspective. And this will be a 3 months double-blind case control study, and this will further be collected at the different time points to better track and monitor the entire process. And lastly, I would like to mention, so eventually we imagine the kind of anti-aging efforts, or the people pursuing Healthspan is more like a entire kind of a comprehensive approach. And we are presenting this more into a more kind of a clinical perspective to help people to improving their quality of health, and also to reverse the traditional kind of approach to sick care, just focusing on the treating the disease, but to instead of that, we're hoping to just pursuing health, and maintaining a good status of the health is more important. So that's the reason we name our longevity clinic as Latipso, which is anadrone of hospital. And eventually we're hoping and working together with our clinicians and all the technology folks we have in the team to providing a more kind of a precise and a preventive care to the our community. And given that, I would like to call back some of the awareness from the field, and also this is another review written by the Biomarkers of Aging Consortiums. So this is more kind of applying the current biomarkers in a more ways to not only applying this as a kind of a side trackers on the current interventions, but also to thinking it more kind of proactively and integrating this more into our daily life, even as some of the, say, the wearables or even your iPhones can help us to tracking some of the data and helping us to understanding the aging process, and also to have the kind of solutions to tackle the accelerating of aging. And eventually at Regenerative Bio, we are hoping to tap in all the kind of original research, all the basic researches together into this kind of translational process, and eventually developing this into a more kind of closed loop, as I mentioned, from the measurements to the intervention. And right now we are building a kind of a global sector among the world. And right now we originally starting our company in Boston, and right now we have a team in the Asia, namely the Hangzhou, and also we have multiple clinical centers in different parts of China and also in Japan. And we have a manufacturing site in the Singapore and Los Angeles, and starting more and more of the clinical efforts around the world, namely here in the Middle East Asia. And this is just some of our stories, because we originally founded our companies in the so-called Gene Town in Cambridge in Massachusetts, and there has been numerous kind of efforts, or the borns of the biotechs, or the even some of them growing into a biopharma. And right now, I think it's time for the traditional kind of a farmer to think about and applying or embrace the world of aging, and also together with the kind of technology development of artificial intelligence. And The other site or the office we have is in Hangzhou, China, and I apologize, the right part is still in Chinese, but you get some of the keywords here is the so-called buzzwords here called DeepSeek. So right now it has been very great efforts and done by like different groups instead of traditional the focus we know at OpenAI or Google. But right now there has been more available and more open access to the AI. So we are not only physically close to the kind of some of the developers or the tech figures on the AI field, but also we are in a more kind of environment and ecosystem to open cooperation and helping this. And there has been some other focuses also have like multiple offices in Boston and Hangzhou, and also there has been some of the like newly founded gaming companies. If you are a game fan, you probably know the Black Myth of Wukong on the Steam, which is very kind of popular and leveraging some of the tech. So my point is, with all this kind of open environment and collaborative environment here, we are hoping to building this kind of AI-driven longevity ecosystem together with the technologies or the ideas minds from the top universities and also the big farmers and also some of the most advanced tech companies. And this is just an overview, and this is the approach we're hoping to achieve with the more clocks or the agent kind of models we have right now, and with the screening design we built, and we're hoping to applying this into a more kind of clinical and future perspectives and helping more people to gain kind of a better quality of health. And with that, I would like to thank all the collaborators and also all the crews from the Regenerative Bio team. And it has been a journey since our launch a few years ago, and right now there's more kind of exciting moments in the longevity field. And this is just our vision, and we're hoping to bring in the health equality to more kind of population and helping people to have a graceful aging for everyone. And thank you. I'd like to have the questions from the audience.
Hello, yes, it's great talk. So I have one question about why you needed the organ-specific, the anti-aging, the substrate or drugs. If we have one, could systematically to get the aging is better than we have more, right? So I just want to understand the behind the logic for this kind of organ-specific anti-aging intervention. Thank you.
Exactly. So there has been two reasons behind this. I think one is you're definitely right. If you have kind of solutions or the so-called anti-aging drugs can helping us to reverse the aging, why not? But right now, the real question is it's very hard to perform this kind of clinical trials purely based on the overall agent, or even have observation on the, say, the extension of lifespan. It's very hard to design or perform or even get like the ASCOs or the approvals from the kind of regulatory perspective. And also, on the other hand, it's very hard to trace this kind of changes on the overall kind of anti-aging process. It takes probably tens of years or even like at least 5 to 10 years to have a very conclusive kind of findings or results to test this. And if you are imagining like 3 different trials from the phase 1 to phase 3, this will probably take decades to complete this kind of trials. And the other is from a more kind of clinician perspective. They have been trained as certain kind of specialties, and they have been understanding the health problems or the existing kind of diseases or conditions at different organs. And they are more helping us to finding or tackling this kind of ways with their own specialties. Say if cardiologists, they are definitely experts on the aging of heart and also et cetera, et cetera. So this will help us to bring in the existing kind of clinical kind of knowledge and also the understandings into the kind of anti-aging processes and helping us to bridging the existing kind of current healthcare systems into a more kind of a future health span focused kind of perspective. Yeah, thank you.
Yeah, I also have one question. Like, because you mentioned for the organ-specific biomarker for aging, and you get that from the blood, and you also mentioned the cfDNA. So, because it's it's DNA, and how you could identify those organ-specific biomarker from the blood?
Yes, so there has been two dimensions of the DNA methylation has been carrying. So one is the organ of origin, which tells you which organs it comes from. And the other is the biomarkers of aging, tells you how old or the current status of the biological age. So both of the information can be extracted from the cell-free DNA, which is already in your blood. And from the more kind of mechanism perspective, as the blood system is circulating around the body, and this has been becoming a mixture of different kind of organ-specific cell-free DNAs already in your blood. The only thing is you need to dissect all this kind of information from a mixture and later ask them like how they can indicate the status of the biological aging of different organs.
So, as I just to make sure I understand this completely, so this is cell-free DNA that you're using, not whole blood DNA.
It's cell-free DNA.
It's cell-free DNA. So, can you help me understand how you're specifically capturing organ-specific aging? Like, what is your way to measure a liver is, say, X amount of age, or brain is Y amount of age?
Yeah, that's a very hard and very good question. So right now, I think there's no definite answer and there's no clear answer for that. And the best way we can do is to have this kind of a longitudinal studies. We have been leveraging some of the public available data for tracing the people for more than over 10 years. And they have this kind of follow-up data, and some of them already have the biological samples before the disease occurs, and with the current status with more kind of aging or diseased kind of organs. And this is more like a benchmark. And the efforts we are building is more like a cohort, and we are helping our physicians or the clinicians to collect all the participants with no diseases, which means they are at least currently medically fully healthy. And this can help us to building a baseline, or at least some of the helping us to understanding the ranges of what's healthy, say, lung or healthy heart may look like at certain age when we have their kind of biological samples. So this is the initial steps, but eventually, or the end game will be if we have, say, a follow-up study for people over 10 years, and we have their biological samples for each year, and we have their, say, medical records. That would be a perfect data set to train the model and fine-tuning this system.
Thank you for a great talk. Wondering if you've compared the performance of your clock with plasma proteomics-based clocks. In recent months, there's been some publications, some coming from the UK Biobank, proteomics data, Olink data, where they developed a biological clock that actually I believe, outperformed the epigenetic clocks that at least were at the time where it was compared to. Just wondering if you've taken a look at that.
Yes, yes, that's another very good question. Right now, we don't have the access to the methylation data for the UK Biobank right now, and it's very hard to comment. And actually, the leading researcher you mentioned for the study is sitting, sitting next to you, Dr. Vadim Glashev. So, yeah. Okay. So the proteomics data and the methylation data, I think they are goes hand in hand, and they are, in my view, are providing different perspectives of the agent. So one is more from kind of the as I mentioned, the switch of the gene and regulates the kind of how the genes turns on and off, and the other, the proteomics, is more like the current snapshot and the status of the agent. And I already read about the publications and also the learn about the data sets. And the proteomics itself is a very powerful data. But if to be greedy or to have a more wishful thinking is to combining these two together will help us to better measure the agent of the status. Yeah.
Thank you.
Thank you.
Thank you so much. So I have a question regarding your clinical trial, the ovarian failure. So can you elaborate more regarding the cohort of your study? Ovarian failure linked to a lot of syndromes and a lot of genetic defects. So how are you gonna incorporate these hormonal replacement therapy, the one you mentioned, in regard to reversing the effect of aging that is induced by genetic defect? How are you going to incorporate that and assess it?
Yes, so I think I did not mention as the current trials, we are more looking for the kind of anti-aging solutions. It's more in the sect of more kind of very mild diseases, or the kind of in the status between health and diseases. So that's the current kind of status we are tackling. For example, we all know there's a so-called term called pre-diabetes, which means your A1C has some kind of on the threshold or tends to be look like diabetes, but you have not developed diabetes yet. And in the clinical trials, people don't to have this kind of interventions in the real kind of clinical applications. There has been different kind of examples. And back to the ovarian health, we are not tackling severe dysfunction of ovarian. We will just enrolling the say the participants with slightly over say the changes on different indicators, say their AMH or the other hormone levels, and they have been reporting this kind of different symptoms. But right now, there has no actual solutions from a more kind of a clinical perspective to actual moving it as a say so-called traditional patients to be tackled. And this is the way we're hoping to provide a more preventive and more kind of proactive solutions for these individuals. Exactly.
Just one follow-up question that I had. So I was going back to one of the slides around validation that you had for the mold clock. So my question is that, so you show that it's better than some of the presently available epigenetic clocks that are out there. One question was, which specific organ clock of yours is better than sort of the presently available clock, since you have multiple organ clocks? And the second one is that the data sets that you showed were for whole blood DNA methylation, but you said that the clock in itself uses cell-free DNA. So I'm a little sort of confused. Was the clock then built for cell-free DNA, or was it built for whole blood DNA methylation? So I'm trying to get those two things mapped. Thank you.
Yeah, thank you. So for your first question, I think right now it's more kind of evolve of the organ agent clock. Right now we are most tackling the well-known kind of organs and systems. Right now it includes 9 different organs in our current version of More Clock, and we are hoping to expand it into 15 or even to more than 20 of the different organs or systems. But with some of the signals, like from lung or from, say, some of the sex-specific kind of organs, it's very hard to get the kind of signals from cfDNA, and that's the reason we might need to consider combining the, say, proteomics or other omics data together. And right now, the current version we are combining is the overall performance of 9-organ More Clock, being called the version 1, and being combining this, the performance with other existing, say, Horvath's clock or Levine clock. And the second question is about, sorry, could you repeat?
The data sets that you have shown seem to be primarily whole blood DNA methylation data sets. That's where all of these clocks have been trained, all the other clocks. But
yes.
The clock that you have built is in cell-free DNA, which is a different kind of DNA altogether. So I'm just trying to figure out how, and requires a different kind of data set altogether. So I'm just trying to figure out how you calculated sort of your metric.
Sure. So the entire approach for More Clock itself is from the cell-free DNA, the circulating DNA itself. And originally, we leveraging some of the understandings from the whole blood DNAs to help us to building or having the initial kind of V version 0.1 or the very alpha or beta version of the model. And right now, the entire kind of model is building on the cell-free DNA itself. And the data has been collected. It's not about like very specific organs, because we are using a more overall approach to collect all the signals from different organs. But right now, we don't have the capability or don't have the enough kind of training or knowledge about the organs we don't know yet. So we're only looking at the organs we know or we have the confidence to looking at right now. That's the current version. But with the gradually we open up the different spaces, and also we have some of the collaborations with, say, certain clinical aspects on especially focus on different organs. We can look into their methylation patterns and helping us to trace back to the cell-free DNA itself to help us. It's more like a kind of iteration process. Any further questions? Yeah, if not, thank you all. Thank you for the staying.
Is it on? Okay. Good afternoon. Thank you very much for spending the rest of your day with me. It's the last presentation of the day. Are you guys joining as well? Thank you. I appreciate it. My name's Nicole Kimes. I'm the co-founder and CEO of Schulte Therapeutics. And we are a company that is founded out of the University of California, San Francisco. We are 8 years in the making, and I like to refer to myself as the reluctant CEO. I am trained as a scientist, and the science was telling us that there's this really amazing thing that we can do, and it's all about prevention of chronic diseases. And what we found was that prevention is hard, and there's some, I would say, skepticism of how we move prevention forward. So it was quite a privilege to be asked to speak here, and I have an admission I'd like to say. I've never once thought of myself as a longevity company. We do really focus on early life and preventing diseases before they ever start. So I was so pleasantly surprised to hear Dr. Mahmood Khan's talk today about Healthspan and really how it's about the quality of life that is going to be so transformative in healthcare moving forward. That is something we are very passionate about and look forward to having a big impact as we move forward. So, prevention, our passion. Why? Number one, let's start with the chronic disease that we're focused on. There's an epidemic of allergy and asthma that's happening across the globe. It originally, when I first started looking into this, it was really considered a Western disease. We now know that's not true. It is much more prevalent in high-income nations, but we're also seeing it in developing nations. It is happening across the globe. So let's talk about the different diseases. When I say allergic disease, I am referencing IgE-mediated diseases. We have atopic dermatitis, food allergy, and allergic asthma. Allergic rhinitis also falls into this category. And what we know is that the incidence of all of these diseases have been increasing very rapidly across the last couple of decades. And as you can see, the numbers I have here on the top are in the United States, and on the bottom, this is in Saudi Arabia. And we have a global issue that is affecting at least 10% of our population here in Saudi Arabia. Both atopic dermatitis and food allergy are closer to 20%. For asthma, I used the overall prevalence, but if you look geographically at different areas, Riyadh is somewhere in the 30% asthma rate. So this is affecting huge portions of the population. In the US, we spend over $137 billion a year simply treating symptoms. This is not even getting at the underlying causes of disease. I looked for the numbers in Saudi Arabia, and I'll be honest, I didn't come up with the exact numbers except for atopic dermatitis, and it was 1.7 billion SAR a year that you're spending on treatment, which roughly translates to 500 million, which is about 1/10 of what we are. And if you look at the population between the two, it's actually quite similar to what we're spending. The way we think of allergic disease is through the atopic march. This is a group of diseases that are Th2-mediated inflammatory diseases. And we see atopic dermatitis typically develop around 6 months of age, food allergy around 1 year of age. Then we have asthma that cannot be diagnosed till about 3 to 5 years of age, and of course you also have allergic rhinitis. Now, we often think of these as developing temporally, and that is true in some cases. In other cases, what we do know is that atopic dermatitis and atopic wheeze early in life put you at greater risk for food allergy and asthma downstream. Those can happen in different orders and in different levels across the board. So now it's often thought of as a comorbidity and less of a temporal march. But in any case, they are related and they are risk factors for one another. So with the rise of all of these diseases, a lot of effort was put into trying to understand what the risk factors were and why we were seeing these changes. There are genetic components to allergic disease, but they describe a very small portion of the disease, and the rapid increase for which we were seeing across the board meant that it could not be genetically driven in predominance. And so there is a consensus that allergic disease is really environmentally driven. And when you look at those large epidemiological studies, the greatest risk factors are things like antibiotic use, antacid use. Most people focus on the antibiotics. They forget that proton pump inhibitors are being used for colic across our infant populations, and those actually have an even stronger impact than the antibiotics. Formula feeding, cesarean section, lack of interactions with furred pets were all of the greatest risk factors that we were seeing. And what that started ticking in our brains as microbial ecologists is all the risk factors are actually impacting the early developing gut microbiome in these infants. So now I'm going to spend just a minute on the microbiome side of the equation. I hope all of you know you are only half human at best. Our most conservative numbers is half microbial and half human, which is interesting in and of itself, but what's really amazing is when you think about the genetic content that means. So microbial communities are these complex, dynamic pools of organisms that are contributing 100 times more genes to our physiology than our own human genetics. And that means that the microbiome is involved in almost every facet of human physiology, including pathologies in which the microbiome is disrupted early in life. We also know that the microbiome is developing very early in life in parallel with the immune system. And in fact, evolutionarily speaking, our immune system is to actually deal with the coevolution of the microbiome system that is part of us. So in healthy infants, you have these very important, we call them often keystone species, which is typical in any developing environmental system. You start with a relatively naive palate. You have these keystone species that come in. They create the environment with which there is further diversification. And that's happening again in parallel with the immune system. And we know that the gastrointestinal microbiome specifically is very important in establishing immune development across the first months and years of life. And that usually leads to a diverse community and a robust immune-tolerant system. So at this point, our hypothesis was that if we're disrupting the microbiome, you're seeing different founding species, and you're actually driving inflammatory responses from the first days of life. So my co-founder, Dr. Susan Lynch, who remains a professor of medicine at the University of California, San Francisco. She's also the director of the Mark Benioff Center for Microbial Medicine, really focused on initially just identifying what risk factors were driving this. And what they found in large infant cohort studies was that there was a microbial signature that they could identify in the first days of life. You saw it through the first 3 months of life. And these infants, based on these signatures, could be put into different risk categories. So in this case, there was one particular group of infants the NGM3 that was at high risk, and they were 2 to 3 times greater relative risk for both allergy at 2 years of age and asthma at 4 years of age. And what was interesting from a microbiological perspective is that, again, they were missing key microbial components in those first months of life, very specifically key bacteria. And conversely, you were seeing an increase in opportunistic fungal pathogens in these infants. Now, when we talk about the microbiome, we typically talk about different taxa and species and strains, but what it really translates into is functionality. What is the functional role that these complex communities are playing? And a good portion of that is occurring through metabolism. You are taking in nutrition, your microbiome is dictating how that is being metabolized, and you are seeing different environments based on the microbiome that you have. So I know this is a busy slide. Focus on blue and green being good, orange and black being bad, and I'm going to just point out one example that's an interesting way to think about how we are looking at different mechanisms of actions, different biomarkers of microbial impact. So in this case, we did untargeted metabolomics in the infant cohorts. We have the high-risk group compared to those that don't develop disease, and we're looking at different specific metabolites. We know that lipid biosynthesis and utilization is very important in immune development, and one of the examples that I'm pointing out here is when you have linoleic acid. So we're going to take the same input for all the babies. It can be synthesized into either polyunsaturated fatty acids, which are thought to be anti-inflammatory, or you get these oxidized lipids that are these dihome peptides. And we can see in the kids that develop disease, the high-risk kids, that they were increased in the oxidized lipids, whereas the kids that remain healthy have the polyunsaturated fatty acids. So we know that it's diet and microbiome together that are stimulating the immune system, but this is a case showing you that even with the same diet, you're going to get different metabolites based on what that microbiome looks like. Now, beyond the metabolism, so what we think we're doing is there's a difference in the microbiome itself. That's changing your metabolic milieu, and then that's impacting your immune system. So for IgE-mediated diseases, this is a Th2 response. You typically see Th2 cells increase, and that's exactly what we see in these kids in the earliest months of life. We can see that their immune cells are already skewing towards a Th2 response, long before you're seeing disease. At this point, it wasn't just our lab and Sue's work that was showing these impacts of the microbiome in IgE-mediated disease, and in fact, we were seeing it in independent global labs. We had Brett Finley's work in Canada, Sue's work in the United States, Hans Bisgaard work in Copenhagen, also another cohort in the US, in the Pasteur cohort. We've since now started seeing these similar findings in both Asian and Middle Eastern populations. And so this is when we started to think prevention might actually be possible. If we can predict it, we should be able to prevent it. So that was the idea we moved forward. So we start with clinical data. We're looking at differential microbial data from longitudinal studies. From that, we're looking for co-associated networks. This is about synergistic functionality. This is not about a single metabolite or a single organism. It is about how do you stimulate an entire community to metabolize food in a manner that is really increasing and supporting immune tolerance. And with that, we do more empirical testing, and ultimately, we need to test this in humans. right? We can do animal studies, and we did do animal studies, and we will always continue to do animal studies. But ultimately, it has to be done in the clinic in humans. So that was our laser focus, was to move rapidly into the clinic. Now, before the clinic, we did do the animal studies. Here's some preclinical data in mouse studies. This is a mouse model of allergic airway inflammation, and this is showing you that based on our rational design of a consortia that was built out of the clinical data that we had seen, in an animal model, you can take a small group of organisms that have synergistic functionality. You can orally provide that, and it provides protection against airway inflammation in an inflammatory model. So this is a model where you use, there's a sensitization period and 2 challenges. We typically use cockroach allergen because this was an airway model, and that is one of the most prolific allergens for asthma. And then we're giving the STMC 103, which is the combination which we refer to, well, the FDA refers to as a live biotherapeutic product, or an LBP. And that was being given orally. We did daily for the first week and then twice per week after that. What we saw was that we do remodulate the gut microbiome from a compositional perspective. We do change the metabolic profile, and ultimately we reduce airway inflammation. So here you can see, you can see from a histological perspective, in the lung you are reducing markers of inflammation. The middle is where you see inflammation. On the top, that's the influx of innate immune cells. On the bottom, that's the influx of mucin, both hallmarks of airway inflammation, allergic airway inflammation. And we were able to alleviate that. When you think about the immunological cascade that's occurring, and again, I'm just showing you one example of the animal models that's for airway inflammation, but this cascade of allergen sensitization, which stimulates Th2 cells, that activates the B cells, they're producing the IgE antibodies, and then upon re-stimulation, you get the whole downstream inflammatory cascade. This is true in atopic dermatitis, in food allergy, and in asthma. It is simply where the downstream inflammation is occurring. That's different. And so in the animal models, we again were able to show with oral supplementation that you can reduce Th2 and you're increasing T regulatory cells, so anti-inflammatory cells. You can see that you're reducing the overall circulating IgE as well as specific IgE for whatever allergen you're using at the time. And also, like many studies, show that you can dampen the downstream inflammatory aspect. So with the animal data, we got really serious about our excitement for prevention in humans. And that's when we decided to start the company, because we found out that most pharma companies weren't really interested in going into newborn babies as a preventative model for a chronic long-term disease. So out of necessity, we built Schulte Therapeutics and decided to do it ourselves. I'm skipping over the whole company generation part of the story. Live biotherapeutic products are an emerging product. We had to build every vertical from the bioinformatics to the manufacturing, every aspect of it. Short story is, we were able to do that. And with the blessing of the FDA, we did a first-in-human study. And this was, rather than thinking about dosing, because when you have a live replicating drug, dosing becomes a very different question. It's also a drug where toxicity is not an issue. So pharmacodynamics and pharmacokinetics that you typically would have to do in phase 1 studies with a small molecule, they don't even apply when you're talking about live biotherapeutic products. So rather than doing a dosing study, we did an age descending study. We did adults, adolescents, and children down to the age of 4. It was for safety, so we treated for 4 weeks, 28 days. Then we did a washout period for 28 days and looked at biomarkers of immune and metabolic impact, and really trying to establish safety to get into infants and newborn babies. High dose adherence in this study, low dropout, no serious adverse effects. Even the adverse effects that we did see, interestingly, out of an abundance of cautions, we had to do diaries for adverse events. So these were not even clinically diagnosed adverse events, and we didn't see a difference between placebo and treatment. Nothing that caused concern here. What we did see, even in adults, and I have to say this was surprising to us, one of the reasons we think prevention is so powerful and necessary is that that's when the gut microbiome is developing. That's when immune system is developing. And we were afraid that by the time we're adults, we have an entrenched system that is not going to be as malleable as what we should see in infants. But I'm showing you data from the adults and the adolescents. And to keep it somewhat short, on the left-hand side, we're looking at beta diversity, which is just a way of looking at the community dynamics of the microbiome. The blue line is the placebo, the orange line is the treated group, and the placebo line, as you can see, is straight, and that means across the study, the microbiome was not changing from its baseline. Whereas in the treated group, in the orange, you could see that as we were treating for those 4 weeks, you're changing the community. And what was really interesting to us is that even after 4 weeks of washout, we were continuing to see changes in the microbiome. Along with that, I mentioned metabolites. We're very interested in the metabolic impact that we're having, so we were specifically looking at those oxidized lipids that are associated with allergic disease. And you can see here, again, our placebo's the gray, orange is the treated group, and we're seeing a decline in those markers of allergy. And then from an immunological perspective, we looked at eotaxin. And this is actually eotaxin circulating in the blood, which was very impressive that we're seeing this much of an impact of eotaxin in the treated groups that we do not see in the placebo group. So safe, tolerable, we're seeing microbial, metabolic, and immunological markers of impact. We went to the FDA and they were excited about what we're doing, and we received fast track designation. There is no prevention of allergic disease at this moment in time. And we continued on to our phase 2 development. So here is the design of our current phase 2 proof of concept study. This is a prevention study, so we are going into an at-risk population. Eventually, we are very much in line with moving towards precision medicine, and we believe that there should be a diagnostic to really pinpoint the microbial predisposition that would be most effective for an at-risk population. However, we need the phase 2 and phase 3 data to develop that appropriately. So our at-risk is clinical at-risk, which is a familial history. So biological, mother, father, or primary sibling is the population. 238 infants have been enrolled in this study. It is powered to see, well, it's powered on atopic dermatitis because that is the first allergic disease that we'll see, and we'll have the greatest incidence in atopic dermatitis. But our fast track designation is actually for type 1 hypersensitivity. That includes atopic dermatitis, food allergy, and asthma. So we are looking at multiple clinical endpoints. We are looking at atopic dermatitis. We are not doing oral food challenges on 1-year-olds in a proof of concept study, but we are looking at food sensitization and clinically diagnosed food allergy. And then atopic wheeze, which is our early indicator of asthma. So we're looking at all of those individually as well as collectively as a single endpoint. Clearly, we're very interested in the safety aspect of this. And so we treat, So we treat daily for the first year. We don't necessarily think that you need to provide these organisms on a daily basis. That being said, we don't know exactly how often they do need to be given, and the clinicians told us, do it daily or don't do it. And that also allowed for the reality of life in clinical trials, and that is people are going to miss their doses quite often throughout a year of the trial. So it's a very conservative dosing that should guarantee that we'll see an impact if there is one. Then we have a year of follow-up. We'll be following the clinical endpoints at both year 1 and year 2, but that second year of washout is designed for two things. One, to tell us microbially, are we seeing engraftment? Are we changing the trajectory of the microbiome in a more permanent manner, or is it a transient thing that we are doing? It's also allowing us to know whether there's durability in the clinical impact. Is this a treatment that needs to be maintained, or are we hitting the critical immunological development period over that first year that would allow for immune tolerance to take hold in a way that is more permanent? I like to point out, I'm often asked why the first year, and there's 2 time points that were really crucial. The first 3 months we know are absolutely crucial to developing immune tolerance. The second time point is when you introduce food. The microbiome changes dramatically and rapidly once you introduce solid foods. And so we were hypothesizing that we would get most people transitioning to solid foods by 1 year of age in the US. So with that, ADORED, Allergic Disease Onset Prevention Study, is the study that is currently ongoing. As I mentioned, all 238 infants have been enrolled. They've actually completed treatment, and the entire study will be completed by Q4 of this year. And I cannot begin to tell you how excited we are to really be able to have the data to back this whole process and really take us to the next step of development of a preventative therapeutic for a chronic disease that has the power to really transform the way we think about development. I put this up here to remind myself. There's a young woman who's much braver and much smarter than myself, who has once told us that we should unite behind the science. We have to take action, and we have to do the impossible, because the alternative is simply not an option. I can tell you this has been a passion project. None of it is easy, but it is absolutely worth doing. And the times are changing. Just in the 8 years from when I first got on a stage and said we were going to do prevention and drug development and was pretty much laughed out of the room, there is now a conversation that is being had. It is possible. It's not easy. But nowadays, I have a few more examples I can point to. One of my mentors before he passed away was John Martin, who was the CEO and board chair of Gilead for multiple decades. And he was my mentor for 2 reasons. One, he believed that scientists should run drug companies, and he was very supportive of that. Number 2, he was the one who brought the first economically viable example of prevention in a targeted population to the market and showed that it is absolutely economically viable to do so. So PrEP for HIV continues to be a billion-dollar market. More recently, we have the GLP-1 agonists. And those speak to a number of topics that we have been trying to bestow on the public over the last decade. And one, prevention is worth it. Two, when you get at the underlying cause of disease, we are going to have greater impacts than we ever knew we could have. It is the antithesis of typical or traditional Western drug development, where you have a small molecule, it's targeting a very specific pathway, and you are knocking down downstream symptoms. When you're getting at the underlying cause, we're really getting at systems biology that's going to have an impact, not just in a single pathway or even a single indication. It's really more a broader impact, and you're seeing that with the GLPs in obesity, targeting all these other chronic diseases, and we think we'll see that across IgE-mediated diseases as well. I would also argue, not only from making money, but prevention is a no-brainer when it comes to saving money. And this is something that Hevolution is doing an amazing job of touting and supporting and really bringing all of the different elements to the table. Yes, we need to develop these drugs so that there is a prevention option, but we have to develop the payers and the government policies and all of the other systems to support a really different way of thinking about healthcare moving forward. This is Dr. Mahmood Khan's talk tomorrow. I will be there, and I would highly suggest anybody interested in prevention be there. With that, I am happy to take any questions. Thank you again for staying this afternoon. Yeah.
You have to choose.
I'm going with you. You spoke up.
Thank you. It was a very nice presentation, and it was something starting from dream to practice. I have two questions. First question, what's the time frame from starting the idea till now?
So depends what starting the idea means. I did my postdoc at UCSF where
well, let's say having the first grant for this project.
The first grant for this project, all right, I'm going to say when we started the company.
Yeah.
That was 8 years ago.
It's very successful.
7.5.
Yeah.
Assuming that we get the data we would expect to see, we will argue for breakthrough therapy, and we should only have to do a single pivotal trial to go through licensure. If that is indeed the case, we've got 3 more years.
Successful story. I have another practical question, which is a debatable question, by the way. I know we can debate on it. But,
Great.
You show that you use animal models to test those microbiomes and all these things. Which animal model? Is it the clean animal model? Specific pathogen animal models, or just normal house animal model?
So we did a couple of, they're all mouse studies. We know that there are lots of issues with translating mouse studies into human studies. So number one, we started with human clinical data, not mouse data. And number two, we moved into humans as quickly as we possibly could from a safety perspective, because I 100% don't believe animal models.
I agree with you.
We've cured cancer. We have cured asthma many times over in animal models, but it hasn't translated in humans. And so we thought it was worth taking the risk. And I'll tell you, this was one of the reasons for starting the company and doing it ourselves. We were told to do a lot more animal models.
Yeah.
And
sometimes
everything.
Several animal models.
Well, and most animals don't actually get asthma. Horses do. Yeah. So at one point, we were told to do horse studies, which is almost harder than doing human studies.
Just to testing the
right, right. So once we really felt good about the safety, and this is an interesting little caveat. In the United States, We could have tried to develop this product as a food and go direct to consumers. It is an emerging field. We are going into newborn babies because we think they have immune modulation properties, and it was clear to us that we needed to do it in a highly regulated, well-controlled manner if we were going into humans. So that was the reason we ended up in the drug route. We also want to cure, prevent, treat, and mitigate disease, and so the FDA thinks we're a drug as well.
Fair enough. Thank you.
Absolutely.
Well, it was an awesome talk, I have to say. I'm a biomedical engineer, so I'm not a cell guy, so I still understood everything you said, so that's really good. And I have to say, and which is not a question yet, that unfortunately I was an investor 8 years ago. I would have invested right away in that company, and that probably would have been a smart idea to do. So congratulations. I know what you're going through with you know, all the challenges that are around and all the questions and all you can't do it, and then you still do it. But anyway, I was supposed to ask a question. So the engineering perspective for me tells me that there is possibly a different root cause for the allergies that develop, maybe the food, maybe the environment, maybe whatever. Have you actually checked that root cause? Is it different in Africa, for example?
So we believe it is the interaction of the microbiome with environmental parameters. That's many different environmental parameters that are impacting
but is
the
the
microbiome
prevalence
itself.
different in Africa or somewhere else where there's maybe a more...
So again, the prevalence is much higher in high income nations. We think a lot of that is because you are actually exposed to more drugs. You are getting more processed food. There's also a pollution aspect. So there's a lot of the environmental factors that interfere with the development of a healthy gut microbiome. Now, I will say, even in developing nations, so India is a great example. They have a low incidence of asthma across the board when you look at the entire population. It's something like 2 to 3%. However, if you look in the children, it's more like 12%. It looks more like what we expect in high-income countries, and we think that they're just behind the curve. And as they develop, they will catch up in what we've seen in the developed nations. Now, one of the questions I get asked sort of towards, are there geographical differences? Certainly from a microbiome perspective, if you just look at the taxonomy of individuals' microbiomes, you can absolutely tell where somebody's from. Where there is less difference is when you look at it from a functional perspective. If you take out the name of the organisms and you just look at the functionality that it is fulfilling, we see much less difference in the developing gut microbiome. And particularly early in life, because it's a very specific, you're going from a relatively naive environment with these keystone species that then develop more diversity, you see even less differences geographically speaking. So a child from Bangladesh is going through that same progression of diversity that somebody in the US is going through over that first year of life. So it was another reason why we really thought it was important to focus early in life when there is more of an overlap globally speaking. Now, again, go back to precision medicine. Could we probably come up with little tweaks along the way? Absolutely 100%, yes. And given the right support, it's what we should be looking at globally to see how we reiterate and optimize on these types of products.
Thank you so much. Wonderful talk. I'm just curious, have you ever tried these in older populations since they have leaky gut?
Any what?
In older populations since they have a leaky gut.
Yeah, so when we started this, I would have, and in fact, I did say no. I said we needed to go into younger, early populations and do prevention. I stand by that in that I think it'll be the most transformative, But I can say even just the small data sets that we got from the adults and the adolescents, I am much more convinced now than ever that we could have an impact as treatment in older populations. So yes, I'm very much interested in doing treatment. I'm navigating a world to make sure that that doesn't overtake the preventative side, but I would love to see both, and we're gonna need both. We're never gonna prevent 100% of allergic diseases. So we need the treatments. There's no doubt about it, and I'm more excited now than ever to do them. Anybody else? Thank you again for staying, I appreciate it.